Thanks a lot.
And also welcome Dr. Nivedita for joining earliest of the meet. Welcome, Dr. Nivedita.
Thank you.
Thank you so much. Yeah, actually there is a post lunch. Just now the lunch time is over and the people are getting ready now. So it's people joining still. Shall we start? Otherwise we wait some more minutes. Dr. Poonam, first session is yours.
I'll be happy to get started actually.
Yeah, okay then. As per the time, maybe we can start. The students will join together along with that. Okay, we proceed for. Okay, I welcome for the day 3 of our international conclave on women scientists in science and technology, with a special reference to biotechnology. So last 2 days of talk, even I see the feedbacks are really amazing, that your limited responses are really great. If people join in the Zoom and also in the YouTube link. So it's a final day and we have some deliberations, and we had a last day the slogan, it's not a competition, but you have proposed some slogan that will be displayed last day. And those persons that are selected persons will be honored today after this talk. So with this, I'd like to welcome Dr. Poonam Thakur. She is Assistant Professor in Indian Institute of Science and Technology, Indian Institute of Education, Science Education Research, Trivandrum, Kerala. And actually, her scientific accomplishment is she is PhD in neuroscience at Punjab University, Chandigarh, and her title on unraveling the molecular pathogenesis of Parkinson's disease, role of heat shock protein HSP and accelerated uterus. And also she completed an MSc in biophysics, and she got a grant of autophagy upregulation of therapeutic approaches for Parkinson's disease, and other outreach grants for the neuroscience, and Welcome Trust DBT Alliance Early Care Research Grant as well. And she had done her postdoctoral research program at Professor John Rober, Coty University, Frankfurt, and also from the Lund University, Sweden. So now presently she is working as an assistant professor at the School of Biology, Indian Institute of Science, Education Research, Trivandrum, Kerala. And she's gonna present the title of alpha synuclein fibril induced disruption of pacemaker pairing in dopamine neurons is dependent on selective K-ATP channel activation. So it's a very big title. Okay, it can be a presentation also very big, but this will be given with 50 minutes of her talk and to share her expertise. And very happy to welcome the Poonam Thakur. It's very long gap we meeting this forum, maybe almost 10 years. We share. I mean, we are trainee of NIMHANS at Bengaluru. So after that, we have very good friendship. Still, we maintaining that. And she's a very great researcher. And apart from that, she's a very good dancer, and also very good fun-filled person. So it's happy to have Dr. Poonam in this conclave. Welcome, Doctor.
Thank you very much, Dr. Mubarak, for having me here. I'm also really happy to come back here. And so I know him since my PhD days, and I can still remember he was a last year PhD student when we had ended the workshop, and the excitement he had for the results and everything we were getting in the experiments of that workshop was really infectious. So that is something I really remember from that time. Let me start sharing my screen, and hopefully it should work perfectly fine. All right, so can you all see my screen?
Yes, but can you go to the slide presentation mode?
Yeah, it is in the full screen mode now.
But it's not, since actually you can press share from the major slide. It's not in the annexure slides. the first slide you have to share. Then only we can see the entire page. Can you stop share and then when you keep sharing that, it will be first one that you have to share screen.
Yes, that is what I did.
But not in the bottom line, the first one. No. But
how about now?
Now, no. Actually, the first one there's in the top one. The screen, not the slide. The screen you have to share.
Okay, yeah. Got that point. All right.
Now,
is it working now?
Yes, it's fine. Please.
Okay, great. All right. As Dr. Mubarak already mentioned, I'm a neurobiologist by training, and my work that I'm going to talk to you about today is in the domains of Parkinson's disease. So never mind the title, it's too technical, it's too specific. Let me just take you through the things, and hopefully by end of the talk, the title will start making sense to you all. And I realize this is a broad audience, so in case anything does not make sense to you or you find anything you don't understand, please feel free to interrupt me or ask me questions at any point you would like. Okay. So what my research career and my lab currently at ISET Thiruvananthapuram is focused on is Parkinson's disease. This is a disease whose name you might have actually already heard. It's a fairly common neurodegenerative disease. So what does neurodegeneration means? It's composed of two words, neuron plus degeneration. So neurons means cells in the brain that conduct our signals and electricity, and where they dying off is what is called as neurodegeneration. And for this particular disease, as like most of the other type of neurodegenerative diseases, we do not have any cure available. So if a person gets the disease, they are doomed to live with it and face the symptoms that come with it. Okay, and Parkinson's disease has a lot of of symptoms. For example, people will have disturbed sleep, they have cognitive disabilities and all, but it is heavily defined by the appearance of these motor symptoms, which means that the patients will have these tremors in their limbs, mainly hand and feet. They will have a stooped or hunched posture. They will be having difficulties in walking. They cannot start movement. They cannot maintain their movement. So basically, what it overall is a lack of movement. So these patients suffer from these motor symptoms. And why do all these motor symptoms arise in these patients suffering from Parkinson's disease? So if you go and look at brain, so this is a picture of a human brain, and this is a midbrain structure. That means somewhere very, very deep in middle of our brain. And these dark brownish colored thing that you're seeing, these are neurons of in the brain region called as substantia nigra. These neurons appear brown in humans due to presence of a pigment called as neuromelanin. And during Parkinson's disease, what will happen is that these set of neurons will degenerate. And these neurons produce neurotransmitter dopamine, which is responsible for maintaining a lot of our movement and regulating them. So because dopamine gets depleted as a result of death of these neurons, therefore patients suffer from all these symptoms I just described. Okay. Now, why will these neurons suddenly start to die off? There has to be some reason, right? So it is usually associated with the appearance of these aggregates of a protein called as alpha-synuclein. Alpha-synuclein is a very small cytosolic protein. It's usually present without much of a tertiary structure. It's what we call as intrinsically disordered protein. That means lacking any fixed tertiary structure. But under disease condition, what can happen is it can really form these cross beta sheet structures, which are highly structured arrangements. And these can form these aggregates, which can gather many other proteins, especially the cytosolic transport proteins and all. And these will form these aggregates, which are called as Lewy bodies, named after their discovery. And these Lewy bodies are believed to be toxic to the neurons in which they show up. However, thing is not really that simple. So these Lewy bodies actually appear in many parts of the brain. So this is if I cut my brain into half and try to look from middle, this is how my brain is going to look like, as like everyone's brain. So here, this light to dark indicates the Lewy pathology severity. That means how much Lewy bodies are appearing in different brain regions during Parkinson's disease. So you see that these Lewy bodies, some brain areas have some small number of Lewy bodies showing up, others have high number of Lewy bodies showing up. So if these Lewy bodies were the only reason for these cells to be toxic to the neurons, then the amount of cell death that we see in Parkinson's disease should also be very similar. But here is the pattern of cell loss, and we see that color pattern in this and in this are not matching. So that means some brain regions are more susceptible to die off in presence of alpha-synuclein aggregates in comparison to the other brain regions. So this is the focus of my research, that what is the reason or mechanism behind higher susceptibility of a particular subset of neurons. And my major focus is these dopaminergic neurons in substantia nigra. So while Lewy pathology occurs in many other brain regions, why is this neuron group majorly targeted during the disease process? Okay. This disease has been described in literature for last 200 years. So obviously, many people before me had this question, and they have been trying to find answers. And in search of answers, few answers that have come along have been something in the tunes of, okay, dopaminergic neurons are very unique. That is why they degenerate during the disease. So what are these unique properties of these neurons? So if you see this, this is a trace of this neuron done in a rat brain. And these are actually the neurons. If you know the structure of neuron, this is a cell body. These are dendrites, and then these will have axons, and axons will further branch out to form synaptic connections with large number of other neurons. Okay, and the axonal arbor or the axon of these neurons are among the largest in the central nervous system. This is excluding the motor neurons, which are obviously the longest because they are spanning pretty much the entire length of an organism. But within the central nervous system, within the cranium, these are the largest neurons that are there. And the axonal branching is among highest, is literally the highest in the brain. Okay, just for comparison, if I on the scale put many other neurons from other brain areas, you can see the amount, the size, and as well as the extent of branching is nowhere compared to the complexity of a dopaminergic neuron. All right. So people believe that because a small cell body has to support such a huge axonal arbor, these neurons are very metabolically demanding. As a result, they tend to die off quickly under disease conditions. Okay. While this certainly describes the differences of dopaminergic neurons from few other types of neurons which face Lewy pathology during the disease process, there are few other things that also define these neurons. For example, most of the neurons in the brain fire when they get some synaptic input. That means a neighboring cell will send it some signal, and as a result, this neuron will also fire. However, these dopaminergic neurons are spontaneous pacemaker firing neurons. That means they are generating their action potentials intrinsically, even in the absence of any synaptic input. They can still fire like this when a synaptic input comes, but even without that, they're always constantly firing. So that means they also have far more additional job to do constantly. So they are never at resting state. That means they have a lot of metabolic burden on them. So, however, there are several other types of pacemaking neurons in the brain as well, which are not affected during disease. Okay. In fact, even within the dopaminergic neurons, so this is a coronal section of the brain. So if I cut my brain in this way, and this is a picture of a rat brain actually, and if I stain here for markers that are specific to these dopaminergic neurons, So this region here is what is called as substantia nigra. Okay, this is another brain region called as VTA or ventral tegmental area. So these neurons are also dopaminergic in nature. They also have this pacemaker firing, and they also have these long axonal projections that I was just talking about. Okay, however, this is a brain atlas picture with the schematic, so that it's easier for you to see. Even within these neighboring set of neurons, that is VTA dopaminergic neurons, they are known to not die off during disease, but just in the neighborhood, these substantia nigra dopaminergic neurons will die off. Even within substantia nigra, the medial substantia nigra neurons, which are closer to these VTA, are more resistant to die off in comparison to the lateral substantia nigra neurons, which have a higher tendency to die off during the disease. But all these three sets of neurons, medial substantia nigra, lateral substantia nigra, and VTA, are dopaminergic, have pacemaker firing, and send long axon projections. So that means there should be some more feature defining the high susceptibility of these neurons. And that is what I wanted to find out. What is that? So to start that out, I thought, okay, let me try to see how, if in what terms we could look in, can we look in their genetic basis, developmental basis, or their functional basis. And we decided to look at their functional basis. That means how their firing changes in the course of disease, or their firing is affected in presence of these alpha-synuclein aggregates, and then try to see if we can get any differences in their susceptibility. Okay, so for this, what we did was I set up a simple assay in which I would study these three subpopulations of dopaminergic neurons separately. But since they are all on a molecular level pretty much same, I cannot use any fluorescent markers or labels to identify one population from another subpopulation. So to identify them from each other, what we rely on is retrograde labeling. That means, yeah. Any question? All right. So what these neurons are sending their axonal projections to these faraway regions, as I just mentioned before. What we can do is, in these regions, we can inject these fluorescent beads, which can travel retrogradely. That means they will travel from the synaptic ends back to the cell body, from here to here in the cell body. And so here are the pictures of these fluorescent ejected beads that I injected in the rat brains. So if and these projections from these different regions are very topologically identified. Okay, so if I inject in this region called as nucleus accumbens, it will send it projections. It is getting projections from VTA. So upon injection here, I will get beads in few days in VTA region. If I inject in dorsomedial striatum, I will get beads back in medial substantia nigra. And if I inject beads in dorsolateral striatum, another region, then I will get my beads back in lateral substantia nigra. Okay, so I did these injections, and after 3 to 4 days, I sacrificed these animals, and then I could obtain the brain slices. These brain slices were then looked at with the help of patch clamp electrophysiology setup. And so what we do here is we take a glass pipette, which is pulled in this shape to have a narrow end. We fill it with some physiological solutions. We bring it close to a given cell. We form a seal, and then we open it up, and then we will have access to interior of the cell, and then we can study its electrical firing or its signals. Okay, and then while we are studying, I applied alpha-synuclein fibrils, which are the aggregated forms of alpha-synuclein, which form Levi pathology, through these solutions. Okay, and when I'm studying them, I can see which are the neurons which contain these beads, which were labeling them. During recording, I fill them with neurobiotin stain, which I can label later, and then confirm that these neurons were indeed dopaminergic in nature by staining for tyrosine hydroxylase, a marker for them. So I'm always very sure of my recorded neurons. All right. So then what I did was then to start recording in these different brain regions. So this is a histological staining of the region VTA, and you can see here these green large ones are the neurons I recorded in this region. And if I look at these recording patterns, just in case you're not familiar with how these recordings look like, or you're not familiar with studying electrophysiological data, I'm sure in textbooks you might have seen these as action potentials. So a spike after spike of voltage change. So this is a resting membrane potential, and then once it's firing, it will reach some threshold and it will go and fire in positive direction, come back, and again action potentials are fired one after another. And this is the time scale. So this is the condensed data of the same thing over 300 seconds or 5 minutes. This is an expanded data of first 10 seconds, so you can see the individual spikes, all right? So when I recorded in this nucleus accumbens dopaminergic neurons, I saw in control solution, neurons are firing happily as we would expect them to. If I apply monomeric alpha-synuclein, that is a native conformation of alpha-synuclein, I do not see any change. And even after I apply the aggregated form of alpha-synuclein into nucleus accumbens, I do not see any change in their firing. Okay, these firing activities can be quantitated through few parameters. For example, firing frequency, how many of these action potentials we are seeing per minute. That is what we have quantified here. We can also look at the resting membrane potential or the average membrane potential here. So there is no change in that. And we can also quantitate the regularity of firing, okay? So this time interval between each action potential, if we just take the average of that and divide, take the standard deviation of that divided by mean of this interspike interval, we can look at how regular the firing was happening. So the lower the value, more regular the firing is. Higher these values are, more irregular the firing is. So we see the regularity of firing is not affected, neither is the membrane potential, neither is the firing frequency. Okay, so these nucleus accumbens neurons are pretty resistant, as we see in the disease cases as well. Then we looked at the medial substantia nigra neurons. Here are some of the labeled neurons that you can see during the recording. Monomeric alpha-synuclein does not cause any change in firing here. But when we apply fibril alpha-synuclein to these neurons, as you can see here, there is some irregularity here and there. But overall, this irregularity was observed only in a smaller percentage of total neurons that were recorded. Many of the recorded neurons, as you can see here, did not show any change in regularity of firing. So overall, it came out to be non-significant. Also, firing frequency overall was not affected, neither was the membrane potential affected. Lastly, I looked at the lateral substantia nigra neurons, which is believed to, which is known in patients to be most susceptible cell population. And here, upon application of monomers, I did not see any change. But when I apply these fibril alpha-synuclein, you can see there is a very quick loss of firing activity. And the membrane potential is also becoming more and more negative, which we call as hyperpolarization. So this is if we quantify them. So you can see a very, very rapid loss of firing activity, as well as decrease in the membrane potential. And there is a massive increase in the regularity of firing here. So that means the pattern of degeneration that we see in the human disease, which lasts decades in patients actually, can actually be replicated in this short experiment of 5 minutes. That means VTA neurons, the firing of VTA neurons show very little effect in presence of fibril alpha-synuclein. Medial substantia nigra, only small percentage of neurons seem to become irregular in their firing, while lateral substantia nigra is pretty much, most of them are highly disturbed in their firing patterns in presence of alpha-synuclein. So that means this pattern can be replicated in these in this short experiment in terms of their firing activity changes. So that means it can serve as a suitable system for studying the mechanism of the high susceptibility of various regions. All right. So in search of mechanism, we looked into various ion channels. So now the action potentials that these neurons are firing, these action potential arise as a result of activity of several ion channels that are present on membrane of these cells. And these include a large number of types and variety, and they are mostly voltage gated. That means they will activate or inactivate according to the voltage of the cell membrane. I'll not go into detail of these things, but what is important is that we know if a particular type of ion channel is affected, what could be the result on firing. Okay, so based on the firing pattern changes that we saw in presence of fibrils, we had our suspect that it is the K-ATP channel, that means potassium dependent ATP channel, that is responsible for causing these changes. So why did we think so? So what happens is that these channels are composed of two types of subunits. One is KIR, or the pore forming subunit, which is depicted here. It will normally remain closed. And another is this SUR, or sulfonyl urea receptor subunit, which is the regulatory subunit. So this subunit will bind to ATP under normal conditions, and it will cause this subunit, pore forming subunit, to remain closed. But in conditions when there is some kind of cellular stress and there is excess ROS production or ATP levels in cells go down, what will happen is that the ATP levels in cell fall down and ADP levels will arise. And then this ADP will go and bind this site and cause the opening of this pore forming subunit. As a result, potassium from inside of the cell will move to outside. And if positive charges are leaving the cell, the inside of cell will become more negative, right? And that is what we had observed in our recordings, that the inside of the cell or the resting membrane potential was becoming more and more negative. So we thought, since this is what is expected of KTP channel, let's see if they are playing any role in causing these changes in firing activity. And to test this out, what we did was we performed few recording in presence of glibenclamide. This is a drug that blocks these KATP channel. It is a selective inhibitor of that. So here we are applying these alpha-synuclein fibrils regularly, and then we perform these recordings in presence of glibenclamide, and we see that the firing loss was completely abolished. And this is what is also quantified here. We can see a very, very clear rescue of the firing loss as well as loss of membrane potential, and firing variability is also quite clearly rescued. Okay, to further test that if KTP channels are really involved in this process, we performed another set of experiments with transgenic mouse. Here we took mouse in where the pore forming subunit of KTP channel was mutated so that it could not open, and we had wild type littermates as controls. And in these controls, if we perform the same experiments, we can see a similar clear loss of firing activity. But if we perform these experiments in KIR 6.2 flox mouse, we can see a clear rescue of this firing activity loss. So that means we, and this is the quantification of the same data, that we can clearly see that in these mouse with mutant KATP channels, we do not see any loss of firing variability or any loss of firing frequency at all. So to summarize my talk overall, it was very clear that KTP channels are clearly involved in this process. And so these KTP channels are believed to be affected by, opening in this case due to most likely some oxidative stress and all. And I'm not including the data here in this presentation, but what some of our other results and what literature says is that when alpha-synuclein fibrils are applied to cell, it usually causes dysregulation in the calcium levels. And these calcium levels can then, excess calcium that enters the neurons can have a lot of devastating consequences for the neurons. For example, they are taken up inside many of these what we call cell organelles inside, especially mitochondria. And mitochondria are, the producers of ROS to a very large extent. So if excess of calcium enters the cytosol, it can also enter the mitochondria through this channel, which we call as mitochondrial calcium uniporter. It is a major source for taking cytosolic calcium inside the mitochondria. And from the data that I have not included in this presentation, we have observed that if we block this mitochondrial calcium uniporter with this drug called as Ru316, we can see quite a rescue of the phenotype as was observed with blocking KTP channels. So that means the ROS production through calcium entry into mitochondria could be one of the reasons through which KTP channels are getting activated. So KTP channels are believed to be more downstream target of this. And if we also block KTP channels, either by genetic manipulation or by pharmacological manipulation, we could see a very, very clear rescue of the firing activity loss. Okay, and with that, I would end my presentation. I would like to thank my lab mates in Goethe University, where this work was majorly done, especially my PI at that point, Jochen Roper, my collaborator, Kevin Luke from Pennsylvania, who provided us with these fibrils and monomers for these studies. And ISO Thiruvananthapuram, where my lab is currently housed for all the funding, India Alliance for the funding. And this is my present lab, and I'm happy to connect with any of you via Twitter, or you're welcome to visit our lab website and check out more about our work. Thank you, and I'm happy to take any questions while you look at this beautiful picture of our campus.
Yes, very thankful to Dr. Poonam for very brief and then very elaborated lecture. So I request the audience can make any questions if you have, and also you can post your questions in the chat box, or else you can directly, you can also request questions. You can, if you wanted to talk, you can just raise the hand, then I can make you.
Okay, no worries. I think that should be fine.
Yeah, I think maybe you're well, like, self-explanatory of presenting all your stuff. So maybe they are. We maybe contact you afterwards.
Yeah, sure.
After this, then you can able to. Thank you so much, Poonam, for your time. And if possible, also be joined for there is a book releasing ceremony also be there after the Dr. Nivedha talk. Be able to, then you can join as well.
Yeah, sure. Thank you.
Yeah, thank you so much. Yes. Okay. Anyhow, formally, I like to thank again Dr. Poonam for an excellent talk on the neuro studies. And next and final talk of today is Dr. Nivedha Sahu. She's a principal scientist from CSIR, Indian Institute of Chemical Technology, Hyderabad. And so to brief about her, so her PhD in marine biotechnology at the University of Delhi and the master program at Ranchi University. She has expertise in algal biology, cultivation, and algal value-added products includes biosaccharide and other molecules as well. Presently, is in principal scientist and CSR IICT, where she works on alkyl-based membrane integrated technology for water wastewater treatment, and also was an assistant professor at the ESCA Academy of Scientific and Innovative Research as well. She has one patent, granted patent, and authored of one book, and four other book chapters she published. Along with that, she received 15 prestigious awards, which include CSR Technology Award, GIYIT Award, CPET Award from various funding agencies and institutions. Her contribution during this pandemic time, where she prepared and developed a 4-layered antimicrobial mask, where he used RO permeate spacer, that got phenomenal recognition for the work. And then she received the outstanding award. She received the BP Pal Memorial Award last year, and she is an state president now for the state of Telangana for the Water Resource Council, that is WSCCI. With a short and brief introduction, I'd like to welcome Dr. Niveta Sahu to give a talk on membrane bioreactors in water and wastewater treatment. Doctor, are you available? You can go to the slides now. Slide.
Hello?
Yeah, you can go to the slide now. You can first. Yeah. Presentation mode, please.
Is it fine?
Presentation mode. Yeah.
Yes.
Yes. Go to, yeah. Fine. Great.
Okay.
Yeah.
So now I'm audible?
Yes.
Okay. Okay, but hello. Very good afternoon, everybody. And today, before starting this one, I must thank Dr. Mubarak for nice introduction. And as we know, I'm at present working in CSIR IICT at Hyderabad at membrane-based intervention on water and wastewater treatment. So with that, today I'll be talking membrane bioreactor for water and wastewater treatment. Before I go, I'd like to give one introduction about membrane. This is actually a membrane is used for various separation technology and highly and efficiently it is used for various industrial like pharmaceutical, food grade, dairy, and cosmetics, agriculture, and many more, even in our laboratories for separating macromolecules or enzyme purification, protein purification, and many more. So, membrane intervention is an important part in industrial process. So, looking that, I'll be talking about membrane filtration technology specifically for water and wastewater. It is an emerging technology which provides very high quality effluent, and this technology has been viewed as an advanced treatment system in compared to the conventional treatment system, which achieves a higher level of standardized water and wastewater treatment depending on its application requirement. So why membrane? Actually, the membrane is basically important on its configuration type, and this slide I am showing how a 3D structure of a spiral module of a membrane, which is showing in this figure. This is showing how the membrane surface, the top layer, and the loading part and other things. This shows this property of a membrane, how it works, which we are working on it to get finer and finer in its hydrophilic property for giving high flux, which pure water, and on a different type of pore size to separate different type of molecular cutoff particles. And this also provides very high maintenance free of system with high quality permeate, zero turbidity, and overall this installation takes very low footprint. The advantages of wastewater treatment is that I already told that membrane intervention takes very low, small size footprint, a very easy installment, high quality of clean and permeate water with a higher removal of organic pollutants and macromolecules, water stability, and with a clean water stability, and many more applications. So this is a simple, I would like to say how the membrane processes, particularly pressure-driven processes, based on different types of the pore, they have been separated: microfiltration, ultra, nano, reverse osmosis, and gas separation. I'll be actually concentrating mainly on nanofiltration, ultrafiltration type of membrane for my this studies, and I'll be talking on this also. So this is overall principle of the pressure-driven membrane process. How here the membrane acts as a barrier between the pollutant and the fresh water, and the principle based on this it works. At last, we have made some synthesis of nanoparticles, and this is different type of membranes which we have made it based on different polymer compositions and nanoparticle compositions for surface water purification. Another, with this study, I am saying that at ISCT we are continuously designing a smart membrane-based technology for water and wastewater treatment, and this innovative and new technology is actually very efficiently treating well to municipality, domestic, and kitchen wastewater. So how does it work? This is a schematic diagram I have shown here that is showing the macromolecules, derivatives, dissolved, uh, 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 soluble and non-soluble particles, organic and inorganic contaminants are within the reactors where microbes are there, microorganisms, either aerobic or anaerobic or algae, any type or mixed cultures. There is different type of are there based on the pollutant, based on the type of the effluent they have been used, and there the membrane acts as a barrier in the reactor to get the clean water, and it retains all the macromolecules thereby. So overall, the bioreactor membrane gives the clean water. The advantages of membrane bioreactors are, of course, no doubt it has a very small, low footprint with very low settlement sludge, high quality permeate or high quality clean water, and high loading rate. But there are certain disadvantages because of what this is not getting that much of success, because membrane preparation is a very tough and tricky process. Membrane falling is a challenging part. Membrane clogging and the pores getting clogged is also a very challenging. Apart of that, the capital cost is one of the hindrance for this process. This is the schematic diagram of the microbe, how it behaves in the reactor, and where we have been working the field solutions, how it is showing, and we are finally getting the treated water. You can say the visible color reductions in this slide. This slide is showing about the how the aerobic, anaerobic in that, and of course algae acts in a different type of reactors. The main purpose to get the clean water, treated water, and of course in compared to the bacteria and algae, they are the bioreactions and metabolic pathways will be varies that I will be talking later. This is overall differentiation between the conventional process and membrane bioreactor, which actually two types we are using in the lab: submerged and side stream. In submerged, the membrane is inside the reactor, whereas in the side stream, the bioreactor is connected with the membrane process to get the clear and clean water. This is the overall schematic diagram that we have been using for municipal, domestic, and industrial wastewater. And at left, we have done at 25 550, of course, 500-liter capacity is also we have treated. This is the setup we are running at the ISCT campus, and we are treating the kitchen wastewater at daily to day basis, and the water is again recycled to the biogas plant, which is getting gas for kitchen, and they are using for tea making and chapati and all making. The schematic diagram of this process is shown, where this plant is actually how it is linking with the biogas plant to get the biogas which is been used in the ISCT canteen itself. And another one, this is the schematic diagram of a plant that is also a side stream of ultrafiltration, which we have installed at LFP, Kottayam Government of Kerala. Continuing to my other studies, case studies with other kitchen wastewater and municipal wastewater. These are the characteristic property of the wastewater, where the reductions of has been found dramatic reduction in color, COD, turbidity, TDS, and other all parameters. And the visible color reduction can be seen in the this picture. Continuing to this, we have done in a coffee industry effluent, and this was also we got this particularly work was published 2020, and the work has been also got appreciated by Vigyan Bharati. Coming to the same thing in a schematic diagram, how the side stream, the sketch it is showing, we are working the lab set the side stream process of the membrane bioreactor, and this is the submerged stream. In submerged, we have used anaerobic organisms, and in side stream, we have anaerobic organism and mixed microbial consortia in the bioreactor, whereas in this aerobic, we have used some microbe we have isolated from the kitchen waste only. Another for getting bioethanol in another study, this is we are showing one stone yeast we have isolated from a rotten tool in the skinning process and compared with other all standards, and we found that particular this yeast was having property to grow in a higher temperature with high yield of bioethanol. So this was identified as Pichia kudrezidi. So now finally coming to algae related work. Before I go, I'll just give a brief introduction. Algae as the autotrophic organism, everyone knows it grows in presence of sun, water, in carbon dioxide, and in return it gives oxygen and of course carbon reduction and many things are happen. During this process, it has been also the biomass which we get, it can be used in various other process like 3F, F which is food, feed, fertilizer. Then the fermentation of this biomass gives biofuel, bioethanol, and many biohydrogen and other things. Similarly, other all chemicals like phycocolors in cosmetics and in food. So in worldwide, among algae, around 221 macroalgal species have been commercially utilized. Out of this, 145 species have been used for food, and 110 species of macroalgae have been used for phycocolor application. But in microalgae also quite important to us. These tiny green organisms are very high in many uses, and this is showing that some species is having high carbohydrate content with and high protein contents, like some examples of spirulina, Dunaliella, chlorella, and many others. Slide is showing how algae is used in various food application and other which is already in market in commercial application. This is of course the food slide which is showing. Of course, in India we are now showing interest, and the researchers are doing many more things, and of course a long way to also go to bring this in practice in our day-to-day life. But this is a usual practice in our Asian Oriental countries like Japan and all. They have been using a sushi and salad and soup in their daily food. This algae, macroalgae, which are also known as seaweed. That other products having in cosmetics, in fertilizers, in biofertilizers, and phycocolors, and many more. Before I go, everybody to this about algae, I wanted to tell everybody that this is actually a daily part of our life. We are using the raw material for algae. For your information, when you get up from the bed, all are using toothpaste to clean our teeth. So the raw material, whatever is used in that toothpaste, it is actually made up from algae. Similarly, some polymers like agar-agar, carrageenan, and alginic acid that we have been using day to day and daily part in our life in different types and different forms that we don't know. Then moving to microalgae. Microalgae, as I saw, this is a microscopy or tiniest organism. Could be a single cell with different type of colony, round in shape or oval in shape or spiral in shape or filamentous, many in shape, but having multiple and different multiporous magnaniferous applications. And they are rich in vitamins, pigments, proteins, carbohydrates, fatty acids, and many more. It is showing overall how the biochemical pathway within the algae. It is from sun. It is taking the process of photosynthesis is happening, and through oxidative phosphorylation, citric acid cycle, the DNA, RNA synthesis, protein synthesis, chlorophyll synthesis happening to final biomass synthesis. Applications, as I said, like microalgae have been used for various things in our mankind, for human food, animal food, feed, wastewater treatment, nutraceuticals, they are rich in beta carotene, astaxanthins, like omega-3 fatty acids, phycocyanins, like highly demanding pigments. They are rich in these beta carotenes and astaxanthins are also highly useful in cosmetics and pharmaceuticals. They are the source that they take carbon dioxide, so they capture and help in carbon dioxide sequestering. They are highly useful in bioenergy as they are being used to produce biofuels, biobutanol, bioethanol, biohydrogen, and they are rich in nitrogen phosphorus, so they have been used for biofertilizer. Recently, algae have been also used for bioplastics, like they are have been found that they are also rich in some polymers like PHA, starch, cellulose. These polymers are very protein-based polymers are actually can be treated and easily it can be converted to biodegradable bioplastics. Pertaining to microalgae and macroalgae that we have worked, we have been found that we are saying that microalgae are aquatic things, but no, we found there are also many species have been reported in air, and these studies were also we published in 2015 where we did the study in ISCT campus itself in different season, and we found that the presence of microalgal species in air. And this is one my work which is showing in macroalgae application in direct cultivation and food. I'll be talking later on. This is biochemical constituents of one it is known as Bradylophia silicina, which shows very rich in lambda carrageenan and edible polymers. as I have, this slide is justifying the algal use in wastewater treatment, which we have done in our laboratory using membrane bioreactor. This is the schematic diagram and how it is showing very clean and clear water. Various species that we are doing cultivation. Another set of experiments which we are doing, how this is used as biofertilizer and fish feed. The fish feed was done in an aquarium tank with ornamental fish, and the biomass was found fantastically increased in 2 weeks. Continuing to some experiments with garden experiments, which shows is the nice for fertilizer with different compositions in brinjal plant. And this is showing again gracilaria, which is macroalgae or seaweed, red algae, which have been used as a co-cultures with prawn at 25 ppt salinity in at 37.5 degrees centigrade, and the biomass of that tiger prawn was found quite uh, uh, high biomass and high yield with good growth rate, like length with 33 centimeter with 60 to 86 per piece gram after 105 days. And the plants were also growing very, these seaweeds were also found growing very luxuriant growth in water. This is summing up with various biochemical parameters just to the audience, how they are having different compositions of protein, carbohydrate and lipids. And these are the various species among Enavena, then Chlamydomonas, Denaliella, Chlorella species, Synadesmus, which can be exploited for either lipids or protein or carbohydrate based on this is the overall we did some biochemical constituents, and it concludes with this and many more. Also it shows that these are the fantastic organisms to exploit for all this purpose for mankind. In another setup we did how that gracilaria in the lab we did tissue culture, and from lab to sea we transferred and it found grown nicely, and from this only I extracted agar. It was tried in with the agar I made some sweet dishes from this, and this was presented in a conference to our institute director and they liked very much. So finally, I can say algae can be used many purposes, and really these can be exploited for mankind for various applications. But there are many more to study and many more challenges are there, like algal cultivation, which needs many development and many research has to be done in Indian water. A part of that, the taxonomy is also needs to be studied critically, and more algal study needs to be carried in coming days. So my questions, whether it is really gives sustainable future, I hope yes, because these needs algae don't need proper land. There is no competition of the land compared to the other radicular plants. These can be grown very easily. Simple culture techniques are there, and the techniques can be easily adopted by the human folks. We have already transferred some technology of gracilaria cultivation at Mandapam Coast and Silka Lake at Odisha. By generating that time during 2010, that people were generating about 5,000 to 7,000 as an additional income for their fishing this profession also. And this algae has been found very eco-friendly, very environmental friendly, helping in carbon dioxide sequestration and reducing greenhouse effect also. So at last, I would say, and about this algae, that algal productions with many more innovative and interesting methods because that needs many more development to get many challenges which has to be answered in like bioethanol sector, then algal cultivation sector, because algal production, it helps in maintaining the 4R circular economy. In my purview, it helps in reducing water. Another are reducing scrap and many in effluents. It helps in reusing and recycling. Algae also comes under reducing air emissions. Along with the biotechnology, with membrane integrated technology with algae, it offers and promises excellent tools for healthcare, food industry, agriculture, animal husbandry, medicinal, industrial processes, and finally sustainable environment management. This also boosts native economy. This also makes our country that recently PM's Man Ki Baat, he has supported like seaweed cultivation to be boosted. So algae also makes Atmanirbhar Bharat. Lastly, thank you. And any questions, I'll be happy to answer.
Okay, thank you, Dr. Nivita, for your excellent talk and detailed information on the membrane bioreactor, the algal-based value-added product as well. So I request the participants, if you have any clarification or question, you can post or you can raise the hand, then I can open, you can ask a question to the speaker. Ma'am, you can also find the chat and I can repeat the questions if you are not able to. So Dr. Sharika Postler is asking some question that she wants some example for the economically important algal species for the organ solvent nanofiltrations.
Yeah, depends what type of actually treatment they wanted to do. So based on the effluent quality, they can do. But good examples have been found. Spirulina, then chlorella, chlorocaina, they have been excellently working for this type of treatment. And this reactor can be later on integrated with the nanofiltration technology to get the clear water.
So next, any other questions? Maybe you can give 1 minute. They can, if they can do type, then we have to wait.
Okay.
Yes. Please. Oh. Ma'am, there is a question from Shakti Pandey, actually suggesting or asking, I don't know. But after the appreciation, he asking whether after the economics analysis of various aspects of biotechnology, she said the additional income of 5,000 to 7,000 is for the month or the annum?
Monthly.
Monthly. Yes, it's
that is that's the figure I have given, which we got for seaweed cultivation at coastal area. Okay, so where a fisherwoman cannot go for fishing, and round the year the fishing doesn't happen when particularly cyclonic air time and raw when weather gets very hard, when it is very difficult to go for in water. Even on other time also, so women back in the family, they can do this cultivation, they can adopt this cultivation things and get additional income. So thereby they were getting 5,000. Now the amount, the figure has been increased.
Yeah. So Shangida is asking how to check the purity of the water after the wastewater treatment. Which parameter is more frequently we can decide the water and wastewater?
See, there are multiple parameters are there, like COD, then turbidity, TDS, color, order is also there. Order and color, which you can do physical like assessment. But these other COD and all things, you can do the analysis. Water analysis, there are so many labs are there. Or turbidity, you can do the simple turbidity meter, which comes in 100 rupees or 200 rupees, I believe, that you can check it. Then COD, of course, you need to go to the lab and you get it done. But certainly, when the gray water or black water is white water, it is quietly known that you are doing. Okay, but it needs to be checked in any laboratory.
Yeah, and Sarika again, she asking that what is the minimum space requirement for the plant for using algal cultivation, especially for the chlorella?
See, depends on the type of quantity of the treatment, how much you are doing, and the type of the bioreactor. So I had shown you like small scale. We did some very small to like 500 that we are doing at the that's the pilot plant scale in our lab, means our office. But you can do with small reactors depending on the generation of the effluent or wastewater at the source. And type of the wastewater also maintains, because different types, like some industries from dairy, the COD and other parameters will vary. Kitchen wastewater, the organic contents will vary. So you have to set the parameters, the water quality parameters, particularly COD, CDS, turbidity. All this you have to see, and based on that, whatever you can do, and then you can go for either algae or aerobic or anaerobic type of bioreactor. Bioreactor should, that is actually providing ambient atmosphere to the microorganisms to grow inside. For that, they can take all this pollutant or nutrients from the wastewater, and ultimately with metabolic breakdown, the clean water comes and that gets filtered with the help of the membrane, and finally you get the clear product. And this water, of course, suppose depends if it is organic, something like kitchen things, you can use for some purpose. But which wastewater that we are using at like cleaning purpose, then washing floor and all these things. So water has been reused; it is not getting wasted. That is another thing. So we are also maintaining zero liquid discharge, that norm.
Okay. I hope the many appreciation messages you could see in the chat boxes, and if they further have any question, they may be contact you through your mail. Okay, thank you so much, Dr. Nivedita, for spending a good time here to sharing your expertise on algal cultivation system and membrane reactor to this our international conferences. So, okay, please stay back. In few seconds, we'll initiate the valediction. We'll also be a part of in that. Can you please unmute and talk? Yeah.
Thank you, sure.
Thank you so much. We initiate the valediction. I'll just confirm with actual persons are available. Yes. Okay. We have come to the end of this 3 days for International Conclave on Women Scientists in Science and Technology with a special references to the biotechnology. So with this, I'd like to give a report of the last 3 days of deliberations. So we had very good, excellent speakers from India and abroad. They have discussed various aspects of science and technology that emphasized on the importance of biotechnology. And we initiated the talk with Dr. Tasneem Fatma. She proposed the title, she's from the Department of Biosciences and of Jamia Millia University. And she delivered algal and cyanobacterial biotechnology and its applications. And they won the second talk by Dr. Renuka Devi from the Sejong University, Republic of Korea. She have explained excellently on how pharmacological aspects of nutraceuticals and nutraceutical based nanomaterial can be used with their potential works and then with recent publications. And the day 2 initiated with Dr. Jyotika Devi from Central Institute of Petrochemical Engineering and Technology, Guwahati, India. She delivered a talk on polymer matrix-based core shell nanoparticles for medical biotechnology, and nanoelectronics-based stuff also she delivered. And the second talk of the day too by Ngoyambak Madeline Nina Lau from University of Douala, Cameroon. She presented on antimicrobials from natural sources against pathogenic fungi. And today and last day we had we chosen the slogans based on the merit into different categories like faculty, student, and research scholars, and also we displayed the top ranked slogans last day. And today we had Poonam Thakur from the Indian Institute of Science, Education and Research from Trivandrum. She presented on alpha-synuclein febrile induced disruption of pacemaker firing in dopamine neurons is dependent on selective KATP channel activations with her expertise on the neuroscience. She presented a very good presentation during our conference. And recently we just had Dr. Niveta Shagu presentation from CSAI Institute of Chemical Technology, Hyderabad. She explained on membrane bioreactor for wastewater and water treatment, and also emphasized on the algal system and cultivation and the different potential applications of the same. So with this, I like to thank all the speakers who participated and consented to deliver a lecture. So we proceed the further deliberations with releasing of the books. Actually, this conference is also be organized by the Nano and Biometal Association of School of Life Sciences. So we take this forum to release our recent book that we prepared from School of Life Sciences, authored by Mubarak and Dr. S. Kamalaza. So this is published by the My Race publisher, Publication Center from Maharashtra. So I can proceed. Okay. So this is our recently publishing our books titled on Brief Facts, it is Nano and Biomaterials. So that's the book title by us. So it's the first edition with ISBN number of that is it mentioned in the top of this book. And these are the two authors. Now you can see that book, maybe overview of this book. This is super fast. So answer. So all the schematic diagrams like this, and the content or the help by some of my students. I should mention their names here. I'd like to thank Ms. Satya, now Mehboob Naushin, Mohammad Adnan, Afan, Ryan for the helping us to make the book came out on time. So with this, the releasing over, and so as I promised, the selected slogan creators that we displayed last day will be honored with our ebook of this one. Will be delivered to your mail ID afterwards. So now felicitation by Dr. Nivedita to give a few words about even the conference, about the book, anything you can share your stuff here. Dr. Nivedita.
Yes, it was very nice. My experience I can share with you. Of course, very shortly we interacted, but I feel the journey is very nice, and I wish you my compliments to you for the book that should reach to more and more readers. And with this, we hope that we will do a lot of things in coming days. So congratulations for this beautiful and excellent conference, and all the students, participants, and co-presenters, everyone. Congratulations. Thank you.
Thank you, Dr. Nivedita. Actually, this is maybe the right person, because she now organizing one book where we are submitted our chapters. So I hope it come out very faster.
I too hope. I hope this year it should come. We are working. Actually, two books are there simultaneously. So again, splitting is happening, and during this process, unfortunately, I lost two of the authors, which is again creating the delay something. I feel very unlucky to have this loss, biggest loss to me. But I am trying from my side to complete their whatever the lacunas are there, editings and some gaps. So these are the extra thing which is taking. And in between, of course, everybody knows COVID time we started. So getting input from people also, that also made delayed and all. So these are the some things which actually make me slow. But I hope this we will be doing.
Yes, thank you so much. Actually, the many participants also been very appreciated your work. Maybe they will contact and do for a collaboration with you in this time.
Sure.
I hope. Yeah, thank you again for giving the felicitation to this valedictory function.
Thank you.
So actually, we actually planned Dr. Poonam was supposed to there, but she's not joined. We have our publisher for publishing our books. The publisher name Dr. Rajesh Thakne, My Rise Publication Center from Maharashtra. Say, I request Dr. Rajesh Thakne to give a few words about this. Rajesh, your voice is not. Maybe you can unmute. It's not muted. Okay. Wait. I think we are not hearing you anything. Sorry.
Hello.
Yes, now it is okay, Doctor.
Yeah. Okay. Yeah. Thank you so much, sir, for inviting me for this function. I would like to thank to all the participants who joined this huge meet, and exchange knowledge with each others. Our vision is to transform each person into the world-class researcher, writer, and publisher to sustain the universe. And our mission is book writing and publication campaign. We promote the authors across globe. We train them, we motivate them, we inspire them, and we give them a pen in their hands. Then they use that pen to sustain the universe. They help society for moving ahead. They solve the problems of society. And we run various campaigns. We are having also a journal named International Journal of Microbial Science, and we publish various books based on microbial science in this journal. I hope that everything will go good in future also. And I'm very thankful to Dr. Mubarak Ali Daud Basha for inviting me to deliver my talk on this occasion. Thank you so much, sir.
Okay, thank you so much, Doctor, for joining us and gave an opportunity to publish our book in your publisher sector. Thank you so much for that. So next, I like to welcome our convener of this conference and representing to felicitate this one. And the book is part of Nano and Biomaterial Association. She is the president of the association as well. So, ma'am, please.
Thank you, Mubarak. I feel that I am really honored to release this book, and also I thank Dr. Mubarak for the opportunity that is given for this book, and I thank.
Wait, பண்ணுங்க. காம்பன் நடந்துட்டு இருக்குல்ல, வந்து நிக்கிறீங்க.
Mubarak, you should mute.
Sorry.
I thank Dr. Sahu and also Dr. Rajesh for joining with us and for delivering the talk, and also I thank all the invited speakers for delivering the wonderful sessions, and I hope all the participants get benefited with this e-conclave, and this is specially organized for honoring the women scientists in the world, and this is for Women's Day celebration and with a special reference to biotechnologists. I hope all the conference like the e-conclave participants enjoyed this one. I hope we can establish collaborations with each other and with the speakers and with other participants also. I hope this will be really helpful. Thank you. Mubarak, unmute. Now we cannot hear you.
Yes. Okay, thank you, ma'am, for your beautiful words. Now we proceed to the next with a vote of thanks by Ranjani, as is a woman scientist, and also the coordinator for Nano and Biomaterial Association of our school. So I request Ranjani to give a vote of thanks.
Thank you, sir. Good evening, all.
Sound is, can you please write the sound?
Good evening, all. It's audible, sir?
Yes.
Yeah. Thank you, sir. Good evening, all. It's my great privilege to deliver vote of thanks for this wonderful event, International E-Conclave on Women Scientists in Science and Technology, with special reference to biotechnology. On behalf of School of Life Sciences, B.S. Abdurrahman Crescent Institute of Science and Technology, and Nano and Biomaterial Association, we thank all the participants for their active participation in this International E-Conclave. We thank the management, Vice Chancellor, the Registrar, Additional Registrar for their constant support and encouragement in organizing this event. We thank BRSI, the Biotech Research Society India, for their association with us to organize this scientific event. We thank all the speakers from various institutions for their insightful talk. I thank Professor Tasneem Fatma, Pro Vice Chancellor, Jamia Millia Islamia, India, Dr. Nina Mandolin Lau, Associate Professor, University of Douala, Cameroon, Dr. Jyotika Devi, Scientist, SciPet, Kolkata, India, Dr. Poonam Thakur, Assistant Professor, IISER, Kerala, India. Dr. Renuka Devi, Assistant Professor, Sejong University, Republic of Korea. Dr. Nivedita Sahu, Principal Scientist, CSIR, IICT, India. So we thank all the speakers for their insightful talk. We thank CAAC Crescent Innovation and Incubation Council for their technical support through providing the Zoom platform. We thank all the members of NBA for organizing this event. We thank My Rise Publication Center, Maharashtra, for publishing our book, Brief Facts, Nano and Biomaterials with ISBN number. We thank all the faculty, research scholars, and students of School of Life Sciences, BS Abdurrahman Crescent Institute of Science and Technology for their active participation and support for making this event a grand success. Thank you all.
Okay, thank you, Mr. Anthony, for proposing the excellent vote of thanks. So finally, I like to thank for your wonderful participation from various parts of India and abroad. For this today, and once when you give a name, please make sure your names are perfect, because with that, whatever the name you are giving, that will be appeared in your certificate. So the certificate will be delivered after arranging all the feedbacks. It will take 2 to 3 days of deliver your certificates. So I can post this link in the chat box as well. So finally, I thank all for joining this. Now the meeting is dissolved. You can note the feedback, and we'll meet in the next good occasion with a good resourceful talks. Thank you so much for all to join.